Hi all of you. This video is about CRISPR Cas9 as a gene editing tool. Now what is this CRISPR Cas9? Now CRISPR Cas9, as if you expand, it is clustered, regularly interspaced, short palindromic repeats, right? And what is Cas? Cas is nothing but CRISPR associated proteins. Now this CRISPR Cas9 system was originally identified in bacteria. And bacterial species uses this mechanism as a defense mechanism to protect itself from the phage DNA. Yes. And recently, this technique is explored in genetic engineering as a gene editing tool. Yes. So let's see how it happens. The history of CRISPR ways back to 1987. In 1987, the mechanism of CRISPR Cas9 was identified in the bacteria E. coli by Japanese scientist Yoshizumi Ishino and his team, right? And afterwards, from 2012, around 2012 onwards, Emmanuel Charpentier and Jennifer Doudna, they explored this technology, this mechanism, CRISPR Cas9 mechanism, and they tried to use it as a gene editing tool, for which they received a Nobel Prize at 2020. Now, what is this CRISPR Cas9 mechanism? How is it used as a defense mechanism in bacteria? This is first a form of adaptive immunity. Yes. So, which means that bacterial chromosome has the memory of or collects the memory of the phage DNA whenever it gets infected by a bacteriophage. So, how does this happen? So it has short palindromic repeat sequences along the bacterial chromosome. There are short palindromic repeat sequences. Now, what is a palindrome? A palindromic sequence is nothing but it reads the same when you read from the 5' to 3' direction in one strand and in the other strands. Similarly, in the 5' to 3' direction in the complementary strand also, it reads the same. Now, there are so many repeat sequences, which are palindromic sequences along the bacterial DNA. Yes. So, in between these repeats, there is something called as the spacer sequence. In between these repeats, there are something called as spacer sequences. Now, these spacer sequences are nothing but they are sequences of bacteriophage DNA. Yes. So, whenever the bacteria is attacked by a bacteriophage, the phage DNA enters the bacteria. A part of the viral DNA is being cut by Cas proteins in the bacteria and it is being incorporated along the bacterial chromosome. Yes, incorporated along the bacterial chromosome. They are nothing but spacer sequences. Now, each spacer sequence is specific to each bacteriophage DNA. So, whenever this acts like a memory. Now, whenever your bacteria gets reinfected by the phage DNA, since we have the memory of that particular viral DNA, we'll have this along with the Cas protein. We use the bacteria used to cut the bacteriophage DNA and inactivate it. Yes. So we'll be seeing how this mechanism happens. But for now, what should you understand? There are short palindromic repeat sequences. In between them, there are spacer sequences. These spacer sequences are nothing but they are portion of the bacteriophage DNA, right? So if you look at this picture, we'll be able to understand the entire mechanism of CRISPR Cas9 which is happening in the bacteria. Now, if you take, this is the phage. Yes, this is the phage. When it infects the bacteria, what happens? The viral DNA enters the bacteria. Now, Cas enzymes. There are so many Cas enzymes. Yes, of which Cas9 is usually being used in the laboratory. But there are other Cas proteins also. So Cas1 and Cas2 in the bacteria used to cut this viral DNA. Now in this viral DNA, the sequence which we are cutting is a protospacer. Yes. And adjacent to the protospacer, we have something called as the PAM sequences, which is the protospacer adjacent motif. So along with this, we cut and this is being incorporated into the bacterial chromosome. Yes. This is the bacterial DNA. So along the bacterial DNA, the spacer sequences, which are nothing but portion of the viral DNA, have been incorporated. It's designated different color because Every time when a different phage attacks the bacteria used to cut a portion of every phage and it used to incorporate it in its own DNA, right? So in between that we also have palindromic repeats in the bacterial DNA. In between these palindromic repeats, we have the spacer sequences which have been incorporated. Now, along the length of the bacterial chromosome, we also have Cas genes and also transactivating CRISPR RNA gene, right? So now when transcription of this bacterial DNA occurs, what happens? Now this spacer sequences along with the palindromic repeats is called as the CRISPR array. Yes, along with this spacer sequences and palindromic repeats is called as a CRISPR array. Now, when this CRISPR array undergoes transcription, a pre-CRISPR RNA is formed. A pre-CRISPR RNA is formed. Now, this pre-CRISPR RNA should become a mature CRISPR RNA, for which what should we do? We have to incorporate it with a trans-CRISPR RNA. Yes, so a trans-activating CRISPR RNA gene after transcription produces trans-activating CRISPR RNA. The function of this trans-activating CRISPR RNA is to link the CRISPR RNA, mature CRISPR RNA with the Cas9 enzyme. The function of trans-activating CRISPR RNA is to link the CRISPR RNA with the Cas9 protein, right? So this is also been incorporated along the mature pre-CRISPR RNA. After which an RNase 3 is being used to cut these pre-CRISPR RNA into multiple fragments, which can be 15 to 20 nucleotide long. Multiple fragments. Yes, so we have so many primary transcripts out of this pre-CRISPR RNA. So why do we produce these many transcripts? Because every transcript, every mature RNA, mature CRISPR RNA will recognize one phage DNA, will be specific to one phage DNA. Yes, so that is why we get multiple mature CRISPR RNA, each being linked with the trans-activating CRISPR RNA. Now the function of trans-activating CRISPR RNA is to move this mature CRISPR RNA and link it with the Cas9 protein. Yes, so these Cas genes will produce Cas9 protein. So this Cas9 along with the mature CRISPR RNA now forms the CRISPR Cas9 complex. Now forms the CRISPR Cas9 complex. Now what is the function of it? Whenever the bacteria gets reinfected by the phage DNA, this is the viral DNA. When it gets incorporated, when this bacteria gets reinfected with a phage DNA, what happens? The phage DNA comes inside. Now the Cas9 protein will go and recognize or sits on the PAM sequence. Yes, PAM sequence, and it allows the CRISPR RNA, which is being linked with that particular Cas9 protein, to check for the complementarity of the viral DNA. Yes, so if that complementarity is established, then what happens? The Cas9 is signaled to cut the viral DNA. The Cas9 is signaled to cut the viral DNA, and then the viral DNA is cleaved and it becomes inactive. So by this mechanism, bacteria protects itself from the phage infection. Yes, the reinfection. Understood? So similarly, whenever other phage DNA whose memory has been incorporated along the bacterial DNA, when it reinfects the bacteria, now the bacteria will have the mature CRISPR RNA linked with the Cas9 protein, which is used for cleavage. Now this mature CRISPR RNA with the Cas9 protein goes and identifies the complementary sequence along the viral DNA. If that complementarity is established, then Cas9 protein will cleave the viral DNA and it becomes inactive, right? So this is what is the CRISPR Cas9 mechanism which is happening in the bacteria. Now, let's have a recap. So Cas9 and Cas2 enzymes, Cas1 and Cas2 enzymes of the microbe will recognize and cut out a segment of the foreign DNA. Yes, after which that DNA segment which have been cut out is incorporated into the bacterial genome as a spacer, right? Now this spacer sequence, after you transcribe, you then link it with the Cas9 protein. Now, when you reinfect, when the phage DNA reinfects the bacteria, the CRISPR Cas9 complexes will recognize the foreign DNA sequence and it will cut it into complete reinfection to prevent the reinfection. Now the Cas9 recognizes the PAM sequence. How this happens? Cas9 recognizes the PAM sequence in the viral DNA. And then the CRISPR RNA checks for complementarity. And then if the complementarity is established, the Cas9 will cut the DNA. Right? What are these PAM sequences? PAM sequences are nothing but sequences with 5' NGG3', where N is any nucleotide. Where N is any nucleotide. Yes, do you understand the CRISPR Cas9 mechanism which is happening in the bacteria? Now let us see how this mechanism is modified to be used as a gene editing tool. So for it to be used as a gene editing tool, first of all, the same mechanism is modified and we cut the target DNA. Yes, so we have a target DNA which is to be cut. So we cut the target DNA using this mechanism, after which the cut ends are repaired by the DNA repair mechanisms. Yes. So what all do you need to cut the target DNA sequence? We need 3 components. One is Cas9 enzyme. Yes, it acts as a nuclease which will cut the target DNA. We have a single guide RNA. So we can manufacture a single guide RNA whose sequence will be complementary to the target DNA sequence. And then we also have protospacer adjacent motif. Protospacer adjacent motif, which is nothing but the PAM. So the PAM sequences which is closer to the target DNA. So your Cas9 will identify the PAM sequence and it will open up the DNA segments, after which the single guide RNA will go and check for the complementarity. If the complementarity of the target DNA is established, then the Cas9 protein which is linked to it will cut the target DNA sequence. Yes, do you understand? This is how a target DNA sequence is cut. Now let's see these few points which we have discussed. Cas9 enzyme acts as an endonuclease that cuts the target DNA sequence. Now what is single guide RNA? There are 2 regions in the single guide RNA. One is a guiding region, the other one is the scaffold region. Now, guiding region is nothing but it's a 20 nucleotide long sequence. The sequence, how have we manufactured the sequence? This sequence is complementary to the target DNA which has to be cut. Then scaffold region is the region which is linked to the Cas9 system, using which it is linked with the Cas9 enzyme. Now, what is the PAM sequence? The PAM sequence is required for Cas9 function, is it not? Cas9 first identifies the PAM sequence, opens up the DNA, after which the RNA which is linked with the Cas9 will go and check for complementarity with the target DNA. Yes, so this is required for Cas9 function. Now this sequence motif is immediately downstream of the target sequence. Yes, so now Cas9 will recognize PAM sequence. What is this PAM sequence? As we have seen, it is 5' NGG3' sequence where N is any nucleotide. Now, Cas9, when it binds with the PAM sequence, it separates the DNA strands of the adjacent sequence to allow binding of the single guide RNA. Now, when the single guide RNA binds to it and checks for the complementarity, then the Cas9 cuts the target DNA. After cutting the target DNA, yes, so we have cut the target DNA. So, after you cut the target DNA, the 2 ends of this the break ends have to be repaired, is it not? So that repair is by the common mechanism that we would have learned earlier, the DNA repair mechanisms. So double-stranded DNA break, yes, double-stranded DNA repair, DNA break repair mechanism is by 2 methods. One is non-homologous end joining, the other one is homology-directed repair. So what is non-homologous end joining repair? First, as you can see, this is the break. Yes, this is the break, double-stranded break which has happened. Now, the Q proteins and the DNA protein kinases will go and sit on the break ends. As it sits on the break ends, it allows the nuclease and your polymerase and ligase, all these enzymes to act on the break ends. So first there will be trimming of your ends. Yes, there will be trimming of nucleotide trimming of your break ends. So after this nucleotide trimming, your polymerase enzyme, DNA polymerase enzyme will add nucleotides and DNA ligase will fill the gap. Yes, so the after which we get a double-stranded DNA without break. Yes, but still, what is the disadvantage of this mechanism? It is mutation prone, is it not? Because there is loss of nucleotides, it is mutation prone. This is one kind of repair mechanism which can happen. Next is homologous end repair. What is this homologous end repair? We are using the homologous pair of DNA. Yes, we are using this homologous pair of DNA as a template, and we are trying to reconstruct the lost nucleotides with the help of the homologous pair, which can act as a complementary or a template strand. Yes, so this is a DNA with a break. This is a double-stranded DNA with a break, and this is the homologous template. Yes, this is a blue color is a homologous template. Now, homologous template, for example, is a sister chromatid. So we also have to keep in mind that not all sequences or this homologous template will not be an exactly identical with that of the break strand, but still there can be changes. There can be changes in few nucleotides along the homologous pair. Yes, but still, instead of loss of nucleotides, or instead of not, there is loss of nucleotides when you consider non-homologous end joining. So that cannot happen in this. So in that case, homologous end repair is beneficial when compared to non-homologous end joining. Yes. Now what happens first? There is a double-stranded break repair. Now this double-stranded break repair. So as you can see, this is the 5' end and 3' end. Along this is the 5' and 3' end. Yes, so along the 5' end, there is some exonuclease activity. So this double-stranded break is then converted to a double-stranded gap with the exonucleases. Similarly, in the other strand also. Yes, so this 5' end is being cut for some extent using exonucleases. Then what happens? So this produces what? Single-stranded 3' ends, is it not? Single-stranded 3' extensions are being produced when you cut out or remove the nucleotides along the 5' ends of the break. Understood? So there are single-stranded 3' extensions, 3' single-strand extensions. Now what happens? The 3' extensions will undergo strand invasion. So homologous template will separate, the double strands of the homologous template will separate, after which there is strand invasion of the 3' end of the break strand. Yes, strand invasion of the 3' end of the break strand. So now that we know each strand has a complementary strand along the homologous template, is it not? So the strand invasion happens in such a way that as it invades, there is branch migration, due to which the DNA polymerase can add nucleotides which can be complementary to the homologous pair. Understood? Then there occurs holiday junction. So as there is strand invasion along one side, along the other side also, the homologous pair opens up in such a way that along the other side also there is strand invasion. So on two sides, when there is strand invasion, there are two crossovers which have been formed. Those two crossovers will be called as a holiday junction. Yes. Now DNA polymerase will extend these two crossovers. Is it not? So these are holiday junctions. Now we have to remove these holiday junctions, or we have to remove these crossovers. Is it not? So the original double-stranded breaks are repaired. So as what does the DNA polymerase do in this case? As the strand invades, it will add nucleotides which can be complementary to that of the homologous complementary pair. Yes. So thereby we have formed two crossovers or two holiday junctions. Now this removal of crossovers can happen in two ways. Yes, one you can remove one you can break the strand which has been invaded, or you can break the strand which is invading, that is the DNA break, wherever the break has happened. Yes, so that is nothing but the orange color. Either you can cut the crossover at the orange color strand, or you can cut the crossover at the blue color strand. Yes, that is what I mean. So after which we will find two strands, two double-stranded DNAs without breaks. Yes, so this can be product set 1. This can be product set 1. So if you break the crossover at the blue color strand, you will get one set of products. If you cut the crossover, or if you remove the crossover by cutting the orange color strand, you will get another product set. Yes, so this is how the holiday, depending upon how the holiday junction is removed, we get different product sets. Yes, so that is what is homologous end repair. What is the final outcome is that we get two double strands of DNA without breaks, completely repaired. Yes, so this is what is homologous end repair. So we have seen two methods. One is non-homologous end joining repair and homologous end repair, which can happen after you cut your target DNA. Yes, yes. So this is what is CRISPR Cas9 mechanism as a gene editing system. First, we use a modified bacterial CRISPR Cas9 system to cut the target DNA, after which the cut ends are repaired by the DNA repair mechanisms. Thank you all.